Hello and welcome to this Future Tech Week session on advanced laser processing strategies. My name is Sarah Kiehne, and I, along with my colleague Erich Prem, represent EIC, a member of the FETFX consortium. This roundtable will discuss the FET-funded projects LinaBioFluid and the upcoming project LaserImplant, both of which look at using lasers to change the properties of surfaces. We are joined by Dr. Emmanuel Stratakis, Research Director for the Institute of Electronic Structure and Lasers at the Foundation for Research and Technology at Hellas and the University of Crete, as well as Project Coordinator of the FET-OPEN projects, LinaBioFluid and LaBionicS. And we are also joined by Professor Dr. Johannes Heitz, who leads the Applied Physics Research Group at Johannes Kepler University in Linz, Austria. Dr. Heitz is the coordinator of the upcoming FET Proactive project, LaserImplant. Welcome to you both, and thank you for joining us. We'll start with Dr. Stratakis. Why don't we start by having you tell us a little bit about your work in the LinaBioFluid project. Please describe how you came into the brilliant idea to analyze the integument of animals.
Yes, actually, this was a very nice project, and we really enjoyed it. So the story starts when Johannes Heitz, our colleague that is currently in the panel, he started some discussions with people that biophysicists in his university, in Johannes Kepler University in Austria, on how we can actually mimic the structures of the integument of the animals, of this kind of lizards and bark bugs that this biologist studied very well, but they have, let's say, studied that they exhibit very nice functionalities on their surface. How to mimic this kind of functionality in artificial surfaces. So he started this discussion, and then we entered in this discussion. We had, let's say, some history on how to mimic the surfaces using the laser structuring. So we use a laser beam to sculpture the surface in order to go down to mimic these extraordinary shapes that the integument of animals exhibit. So we have started this discussion. We have applied for this project, and actually it was a quite successful project. I can recall that at that time the percentage of the rate, let's say, of success was about 2%, close to about 1.7%. So it was a really successful project. So the main idea of the project is to mimic these structures in order to manipulate or to control the fluid transport in artificial materials. So the nature showed us the way. We have studied this way, and we have managed to mimic in order to produce artificial materials with these extraordinary microfluidic properties.
Great. So please describe a little bit about the LinaBioFluid project in terms of technological and scientific impact.
Yes, LinaBioFluid actually, through this process of mimicking nature's, let's say, strategy to manipulate liquids, has managed actually to provide artificial materials that show exceptional friction reduction. So imagine that we have a slide bearing in a machine. Then if you treat this slide bearing with a laser processing that we have identified through LinaBioFluid, then this piece of machine shows exceptionally reduced friction. So this means actually that there is a reduction in the energy consumption, and in the long term, a reduction in CO2 emissions. So this would have a great impact actually in the current technology of machines. I mentioned one of the examples, but there are plenty of examples for example, mechanical components in microfluidics or in micropumps, that if you treat the main part with this surface finishing, the surface processing, then this will show exceptionally reduced friction. So this is very important, because if you imagine, friction is one of the biggest problems of the most energy consuming issues in the modern technology. So if we reduce this friction, we reduce the energy consumption and then the CO2 emissions. So this has a great impact in currently.
Great.
Great. Sarah, can I step in with a question?
Of course.
Emmanuel, that's a fascinating project. I was wondering, I mean, this is an interesting combination of let's say a science project, which is the bio part, then the physics part, which is obviously the laser and the surface physics, but then also a technological one. What came first, and can you describe a little bit? I assume you also in the project had to look into some inspiration from science. How did you do that? How did that work?
Yes, everything has, I mean, this is mostly a basic science, has started from the basic science problems. So the biologists have, let's say, have the very fascinating problem to understand how nature is working and how through the evolution these functionalities has been developed by nature. So this is the one part. The second part is how to fundamentally study the laser material interaction in order to control the shape on a material surface, the geometry, the size, the periodicity. So in fact, it's a fundamental problem of laser material interaction that can give you this capability to control, to precisely control the shape. So this combination, so this is an interdisciplinary, let's say, interaction, and that's the highly disruptive, let's say, idea. If you combine this expertise, these two fundamental sciences, then you end up with a great technological, let's say, invention. And this was actually the idea that that LinaBioFluid entered into the table. So that's, I mean, it has all started from basic science, and then this evolved into technological, let's say, invention.
Great. So Professor Dr. Heitz, why don't you tell us a little bit about the upcoming laser implant project? What are going to be the aims and expected outcomes?
Maybe I go here a little bit in history.
Sure.
So the LinaBioFluid project, we had as animal models lizards. This was a little bit older, and a new idea which started in principle with LinaBioFluid was bark bugs. So bark bugs are bugs, really bugs is some kind of insect which live on the bark of trees, and they have the possibility to guide fluid quite rapidly. And we thought this could be a good starting point for the reduction of wear and friction due to the guidance of oil. Mm-hmm. And we, as Emmanuel described, we succeeded to do it. And we could indeed measure reduced friction with contact between laser processed surface which had similar features like you find on the bug, and steel counterpart. And the material which worked best at this time was a titanium alloy, which was processed by the people, the project partners in Berlin. And parallel to it, we had some activities where we tried to reduce the adhesion of cells, biological cells, on implant surfaces. And the background was that we had a cooperation, still have a cooperation with people from our cardiology department in our university hospital. And they implant pacemakers into the heart, and they have to be removed after some years because of battery lifetime. And they should not be totally overgrown by tissue. And the idea was whether you can change the surface properties that they don't get overgrown. Mm-hmm. And it was the same material as we had for the wear reduction. And I thought, let's take the samples from Berlin and let's test how the cell react on them. And indeed, we found that also the cell adhesion was reduced at exactly the same material. So we, in principle, developed the material for wear reduction, but we found out that at the same surfaces, we see also cell reduction and a cell growth reduction. And based on this, we started an innovation launchpad project with a smaller budget, with only a few partners, with the partners in Berlin and the hospital. And we tried to find potential applications for this idea. And we succeeded to find some companies who would be interested to use it and to test our samples. And the most easy and the most promising thing was bone screws. So with bone screws, it's the same material, also titanium alloy, and they are screwed into bone. And also most of this, and nowadays most fractures in bones are treated with these screws, and also these screws have to be removed. So typically, maybe not the screws themselves, but they fix plates onto the bones, and the plates have to be removed. And to remove the plates, you have to remove also the screws. And if you wait too long, and this is half a year or something, then it gets difficult, and the screws can break, and then the minimal invasive treatment, which is with these bone screws and plates, becomes not minimal invasive anymore. You have really then to cut out the screws. And so we came to this point that it would be also interesting to have bone screws which have these properties. And during this launchpad process and a little bit later, we then found out that it is possible to get cell reduction also on screws. And it was a little bit more difficult because it's different cells which are of interest. So typically the pacemakers, the cells of interest were fibroblasts. Fibroblasts are cells of the connective tissue. They produce a lot of collagen. They make scar tissue, for instance. And for the screws, it would be osteoblasts. These are cells which are built up bones. And they were more difficult. So they liked the surfaces, rough surfaces more because they live on rough surfaces in the bone. But also here we found parameters that we can suppress the cell growth on the screws. And then we decided, and we were offered, of course, and supported by the European Commission, and they announced that there is a new kind of projects, this Pathfinder projects, supported by the, I think, European Innovation Council, and which exactly has the idea to bring out results of FET projects to a higher technology readiness level. And we thought about it, and we talked with the people from the company, with the bone screw company, and we also came in contact with other people working on dental implants where you have similar things. Maybe there you would like to have a better integration into the bone, but it could also, this effect can be reached by laser processing with a little bit other parameters. And this was a French group, and we put them all together and then applied for the laser implant project and were successful. And we will start in 1st of January.
Great. So as far as how you would describe the innovation potential, potentially resulting products or services?
Yeah, so for us, it's clear, it's implants. So dental implants or bone screws. Of course, it could then also, maybe we could come back to the original idea again to have smart, for instance, pacemakers for the heart. But this is more complex. So the whole system is much more complex, and we also somehow with the surface treatment, we intervene somehow with the function of the implant. So we decided to go to the simpler systems first, which are mainly passive. But this could be extended if we are successful.
Maybe can I ask a follow-up question also to Emmanuel? I mean, you mentioned friction and bearings as potential targets. I guess, I mean, this is a huge, as you said, this is a huge application area. I mean, is this something that you were targeting at the start of the project, or is this something also that developed a bit more during the project?
This was the main target of the project.
Right.
So we have promised actually a very high percentage of friction reduction during the writing of the project. And unexpectedly, we didn't expect that we will manage to reach such a high friction reduction through this process.
This is actually, this brings me to a follow-up question. I mean, the EIC Pathfinder and previously the FET projects, they are a little bit special in that you have, as you called it, basic research, or let's say a more long-term research perspective, but you do have a technological objective in mind. So some researchers, or at least some research administrators, think there is a tension between these two aims. But we have now also talked to some other researchers who say this is actually not a tension at all. This is more kind of aligning research. How do you see that? Is this, how do you perceive these, let's say, this double challenge of doing very good long-term research, but also having a technological objective?
Yeah. Let me first say that everything is started from the basic research. Nothing can be ordered, and anybody that says that we today will order, we will identify, let's say, will invent, for example, the laser. This is, I mean, this is not, let's say, a good, the correct, let's say, thought of research. So everything is started from curiosity-driven research, and then you build up on this research when this kind of research is mature enough in order to produce these technological advancements. So behind this technological objective we had in LinaBioFluid were many years of basic research on laser material interaction, as I mentioned, and also on the biophysical models that we had in mind. So I think that the main, let's say, there is no a dilemma to my perception. We have to invest on basic research, and then through this process, we'll end up with highly technologically important inventions. Otherwise, we will be we'll make, let's say, an incremental, let's say, incremental technological inventions. The big inventions start from investing in basic research.
Right. Maybe Professor Heitz, would you like to?
Maybe I can also comment a little bit on this. Also, let's say with the example of the LinaBioFluid. So actually, we as people at the university, we go for both. For us, of course, also the basic research results are very important for ourselves, for our careers, for the careers of our PhD students and of our postdocs, which are working in the projects. But we had from the biology, which came mainly from the university in Aachen, and my colleague Werner Baumgartner, who was former professor in Aachen and now changed here to the JKU again, to the Johannes Kepler University in Linz again. He brought up this idea as an expertise in biology, and they have made quite good things over years, and they have especially started the water transport of desert lizards which on their skin transport the water to the mouth. And they have made some theories and they thought they understand it. And they also worked already with laser processing to mimic these things. And so we had some new idea with these bugs, which also have a very specific microstructure for liquid transport. But we were quite confident that from the basic research point, we will be successful because we have already such a huge experience. And this would probably, if we would probably be able to understand the principles and probably will be able to mimic the structures. But then we had also in mind for what could this be used. And then we came for wear and friction reduction. And this was not true at all. We had nothing in this direction. And we thought, let's try it. And the people in Berlin had worked a little bit in this direction, but not with the structures. Let's try it. And so this was the risky part, I would say. The basic research part, yeah, we continued our ongoing quite successful activities. And the risky part was then the application part. And it also came up in the project, as I described, that the applications which we thought at the start was not the only potential usage, but we found other things as well. Emmanuel also will describe, certainly they had quite nice results with anti-reflective surfaces. And so we found other applications as well. And we just went in with one target, but we also with open mind for application. I think this is the main point. You have to start from a good base on fundamental science, but you should be open-minded for potential applications. Yeah. And the FET projects give a good base to do this.
Can I come back in the? So Johannes is right. I mean, there is an endless potential of this, let's say, technology that has been developed through basic science in many sectors. For example, we have targeted through LinaBioFluid friction reduction, but then you can see that, let's say, biological adhesion entered into the game. Also anti-reflection, as Johannes mentioned, we have developed an anti-reflection technology based on the same strategy to mimic the nature's, let's say, strategy on how to produce anti-reflective surfaces, and to push forward many ideas, many other ideas through this process. So it's quite, let's say, fascinating to start from your knowledge and see that there are many windows, many opportunities of technological advances.
You briefly mentioned it already. Maybe I can have a follow-up question here, and this is the nature of the FET program, or the EIC Pathfinder FET, as it's going to be called from now on, which aims for precisely what you have described, long-term research, but with a technological long-term perspective. I mean, what do you think is special or important in this program? What would you say are the aspects that should be maintained in the EIC also in the future? Or maybe we start with you, Emmanuel.
That's a very nice question. It's my favorite. Since, I mean, it is a lot of, we have a lot of experience from this. We have started from FET, of course, the FET-OPEN, but through this, the different, let's say, tools that the European Commission provided us, I can, let's say, I have a very clear, let's say, opinion on what I think that it will improve, what I think that it is very nice that it is present now. So the European Innovation Council, let's say, calls, the current European Innovation Council calls are quite helpful, I can say, for a researcher that would like to push forward a technology. So let me start from LinaBioFluid again. Then we have these two three actually FET innovation Launchpad projects out of this project. LaBionicS, that is targeted to has targeted to under-reflection technology. The one of the cell-free implant, as I can recall from Johannes and his team, that is targeting biological adhesion. And also there is BioMarble, I cannot recall it very well, but it's an initiative from the European Institute of, the Spanish Institute of Optics, led by Jan Siegel. So, and is targeting actually how to produce improved surface finishing of marbles. So there are three technologies came out of LinaBioFluid. Then Johannes has created, pushed forward another European Innovation Council tool, that is the FET Proactive tool, and push forward the TRL, the technology readiness level, for his research, for his concept. And we also, through LaBionicS, pushed forward an anti-reflection technology. We are currently have funded by another tool of European Innovation Council, and this is the EquiFund, that is, let's say, cooperation of the European Investment Bank with the Greek government in order to finance startup spin-off companies that came out, let's say, from this from European Commission competitive calls. And we are very happy that we have initiated the spin-off company named Biomimetic that has just launched, and we have attracted the seed funding of about 900,000 euros. Okay, I wish I because now anyway. So this actually completes a circle that has started from a basic research, and then we have exploited all the tools of the European Innovation Council in order to push forward our technology. But there are a lot of, let's say, there is a lot of room for improvement, and I strongly, let's say, support the idea of investing more money following this kind of basic research proposal, like ERC or FET projects. Unfortunately, there are not many tools to fund basic research, but at least those we have to invest to those people that came out of these projects to help them push forward the TRL. Also, I have written this also in a forum of the European Innovation Council. I strongly support mono-beneficiary tools that will provide the researcher that came out of this project to push forward his idea, his patent. So if he has a patent out of these projects, then he can take the resources, he can receive the resources in order to push forward this technology. Because there are some, let's say, difficulties if you would like to have to interact with the current industry. There are IPs, for example. The researchers do not want to share their IPs with industries. So it is sometimes difficult to interact with big industry in order to push forward the technology. So if you are the only beneficiary, then you have all the measures to push forward your idea, to make, let's say, to push forward an SME, or to start an SME, to build your SME, and then to push forward your technology. So this is, to my opinion, the environment that a researcher faces from the writing of the basic idea towards the market.
I don't know, maybe Johannes, would you like to add what your favorite aspects are of the program, and what should be maintained in the EIC programs?
Yes, I also like the ideas that you can really follow up the results from the basic ideas, which are very free, which is also very good. it's thematically open, which I also like very much, because it gives the room for wired ideas, let's say it like this, and which have nevertheless often very good outcomes. Because if you always go on the same path, then maybe you will be very successful on the long run, but some wilder combinations of researches and ideas often give quite interesting results. And to have the possibility to push them further, it's really a good idea. And also what we did now is, also in the LaserImplant project, we had inclusion of SMEs. We did not found our own SME. Actually, our French partner had SME already, which was a startup from the University for Laser Processing. But to include especially SMEs in this raising of the technology readiness level, I think, is also very important.
Sarah?
Great. Well, I think that about covers our questions. So we would like to say thank you very much to both of you for taking the time to talk to us today, and we wish you all the best with your future research endeavors.
Yeah, same from my side. Thank you very much for the discussion.
Thank you also very much. It was a great pleasure, and it is very nice, let's say, initiative to discuss with researchers and people that they strive a lot, let's say, to receive funding, but yes, but at the end of the day, to produce something that it is very useful for Europe, for European citizens.
Yeah, absolutely.
I also thank you very much for the possibility for us to present for broader public our projects and our ideas. And I also hope that this ideas of the strengthening of the innovation in the European funding continues.